Our last video for this chapter focuses on immune system disorders. And so what would constitute a disorder is if the immune response is too much, too little, or directed against the wrong targets. And so we'll talk about too much. Too much of an immune reaction, an excessive harmful immune reaction to antigens is called hypersensitivity. And this is the largest category because it includes allergies. So reactions to environmental antigens, also called allergens, the common ones that you're familiar with: dust, mold, pollen, vaccines, stings, poison ivy, foods, drugs, kind of a long list of what we can be allergic to. But what happens is that your body or immune system has an overreaction. And then your book goes into classifying these into types. I really don't know that we need to go through all of that, so I'm going to skip. And I'm going to get to, past all these types, I'm going to get to what constitutes an autoimmune disease. So an autoimmune disease, if the self-tolerance is failing. So if the immune system is not able to tell self-antigens from foreign ones, that's considered an autoimmune disease. And so some immunodeficiency diseases is if the immune system doesn't react enough. So too weak of a reaction, and some of these you may be familiar with. So SCID is when there's a lack of T and B cells genetically, so you did not inherit enough. There's not directions to make enough T or B cells, and so this individual would be vulnerable to infection and would have to live in a protected enclosure. Probably your most common or most well-known immunodeficiency disease is AIDS. And AIDS stands for the Acquired Immunodeficiency Syndrome. It is not genetic, and it can be contracted after birth. So this is a group of conditions that involves and severely depress the immune response. So the AIDS is caused by an infection from HIV, which stands for the human immunodeficiency virus. And so this virus is a retrovirus. We'll get to what that means in a second. But the virus invades the helper T cells and macrophages and basically tricks them to internalizing the virus, and then they kind of get to work. So this is a picture of the host cell and then inside the virus. So a virus, all a virus is some genetic material, usually RNA, with a protein coat, which is a capsid. And then what you're seeing, the rest of that is the cell, the host cell. And so let's talk about how a retrovirus works. A retrovirus means they use RNA from a virus, and that is the template to make DNA. Now, back in A&P 1, you learned that DNA is used as a template to make RNA. So that's why it's called a retrovirus, because it's going backwards. So the RNA is used to make DNA, and then that new DNA that has the information from the virus is inserted into the host DNA. Now, this could lay dormant for months to years, but it's in there. And so when it's activated, it induces the host cell to make more viral RNA and proteins. And so the instructions that are on the new DNA are to make more viral RNA. And so that viral RNA and that whole process destroys those helper T cells, and so that allows HIV to strike at the central agent of nonspecific defense, and it kind of doesn't allow those helper cells to help with cellular immunity. So looking at some signs and symptoms of the HIV virus: flu-like symptoms, chills and fever, night sweats, fatigue. And then if you have HIV, you would see your helper T cell count lower. So a normal person is from 600 to 1200 helper T cells in a microliter of blood. But with HIV, you would expect that to start diminishing because those viruses are essentially destroying those helper T cells. But to be considered having AIDS, it would fall all the way to below 200 helper T cells per microliter. So that's kind of the cutoff there. And then this talks about some other possible infections because the immune system has been diminished. You're more susceptible to other infections. So ways that the HIV virus is transmitted. When this first became emergent, it was very confusing as to how HIV was transmitted, but it is transmitted through bodily fluids. And the most common means of transmission would be sex, contaminated blood products like in a blood transfusion, or needles, but it is not transmitted by casual contact. So we now have some treatment strategies for AIDS patients or patients with HIV. The treatment prevents binding of the proteins to the helper cells, so it kind of stops that process, and then it also disrupts the new viruses from making the DNA, inserting it into the host cell. So of the medications that are available, none of them are able to eliminate HIV, but they are developing resistance. And so AZT was the first anti-HIV drug, which inhibits that reverse transcriptase, making the RNA, or using the RNA to make DNA. But now there's more than 25 drugs on the market to help with slowing down this virus.